This Week in Virology, the podcast about viruses, the kind that make you sick. Welcome to TWiV. Right. Thank you. From Microbe TV, this is TWiV, This Week in Virology. This is episode 464, and today is October 18th, 2017. This episode is sponsored by the Defense Threat Reduction Agency, part of the U.S. Department of Defense. The agency's Chemical and Biological Technologies Department hosts the 2017 Chemical and Biological Defense Science and Technology Conference to exchange information on the latest and most dynamic developments for countering chemical and biological weapons of mass destruction. Find out more at cbdstconference.com. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. We are coming to you today from Boston, Massachusetts, a city where I spent 3 years in the late 70s. And we are actually in a place I'd never been before. We're at Tufts University. We're in fact at the dental school. It's a little scary. We're on the 14th floor, and there are these stations behind us where you learn how to drill and do things to teeth. I have to say, dentists were not my favorite doctors. It's a little scary, but we're okay. They're actually having a class right now behind us. And we're here courtesy of the Boston Student Postdoc Chapter of the American Society for Microbiology. And I have garnered 2 virologists from Boston. It's not hard to get virologists in Boston. There are plenty here. And so this is a subset of all those present. All the way at the end of the table from Tufts University, Katya Heldwein. Welcome to TWiV.
Thank you.
Did I pronounce your name Heldwein?
Correct, yes.
Like held like a wine. Glass of wine.
That's right.
Perfect. And on my left, from Harvard Medical School, Sean Whelan. Welcome.
Thank you.
And Sean, I've known for many years. Katya, I've met you for the first time today. And that's the way science goes. And we're going to talk a little bit about their histories, how they were trained and so forth. We'll talk a little bit about their science. And then at the end, I have some tricky questions to ask them. So that'll be the fun part. Which they don't know about. And so they can't say, I don't know, right? They have to give an answer. Let's start with Katya. Tell us, where are you from?
I'm from Moscow, Russia.
Moscow.
Moscow.
And when did you go to college there in Russia?
Yeah, so I was I got my undergraduate degree in chemistry at Lomonosov Moscow State University. Yeah, and so, but I was really more interested in biology, and so I decided to apply to graduate programs in the United States, which was quite hard coming from Russia, and a lot of programs that I wanted to apply to required GRE, not only GRE general, but also GRE subject in biology, and I just never really took biology. I think the last biology course I took was in high school. So I was happy to take chemistry, but I couldn't take biology. So sort of like my, where I applied was very much influenced by what were the restrictions. But anyway, so I just kind of want to share a funny story. So when I was applying, I had to fill out the major and minor descriptions. And so, I mean, I studied English, but I studied British English, Oxford English. And so I just really didn't come across this major and minor concept. But of course, I had some understanding of English, and I thought, well, major must be something that you study a lot. Then minor must be something that you hardly study, right? I mean, it was logical, and so I thought, okay, major, and I thought, okay, and I think depending on the place I applied to, I put either organic chemistry or physical chemistry, and then I had to think about what was the course that I just basically was just so marginal in my education, and I realized that it was political history of the Communist Party. So that's what I listed as my minor. It was only after I came, and so needless to say, I didn't receive very many responses from universities, perhaps because of that. But only when I moved here did I realize my mistake.
Where did you end up going here?
So I got my PhD at Oregon Health Sciences University. I was in the biochemistry department, the graduate program there. And that's where I got introduced to crystallography and really became a structural biologist.
So did you go there with the idea that you would do structural biology, or did you?
I had no idea what structural biology was. I had no idea what molecular biology was, what structural biology was. I mean, I sort of understood biochemistry because I took physical chemistry. Yeah, so so taking biochemistry the first year was easy, relatively easy, because I understood the chemistry and the physical chemistry behind it. But I remember the molecular biology course was absolute murder. I remember the very first lecture in it. It was just, I guess it was already like advanced level course, like students were expected to know molecular biology. And I remember it very vaguely, but it was taught by the late George Crossa, a very famous microbiologist, and he was teaching, I think, from what I recall, it was something about plasmid replication, and he was talking about these plasmids kissing each other, and I sort of have heard of plasmids, I sort of knew what they were, but that was about it. It was really, really hard course.
Is the kissing part that you didn't quite get?
And he was so excited about it, and it just, I guess, like the fact that he was making that joke didn't make it any easier for me to understand the process.
Did you then do a postdoc after that?
Mm-hmm. Yeah, so after that, I decided to continue my training in structural biology, but I became very interested in viruses. And so I came to the lab of Steve Harrison, who published a lot of, so he's done a lot of research on viruses, specifically structural biology of viruses. And so I came to his lab, and that was a very exciting experience. And so it was there that I started working on herpes viruses and continued.
So did you, again, you probably didn't go wanting to work on herpes viruses, right?
No, I really didn't know anything about herpes viruses.
But when you got there, he said, these are interesting, you should work on them.
No, actually what happened, so I was supposed to work on the project that he pitched to me and I agreed to work on was actually entry of dengue virus. But when I started, it turned out that there's another postdoc working on it. And I guess like Steve either forgot about it or he just thought that that postdoc wasn't really doing anything. So anyway, and so it just became really awkward, and it was clear that we couldn't really divide the project. And so, when I was in graduate school, I liked working on multiple projects. And so right off the bat, Steve gave me a second project, didn't really have much, well, had something to do with viruses, but indirectly it was working on clathrin adapter complexes, which of course are involved in endocytosis, which viruses use to get inside the cells. So it is connected. So I was working on it for several years, and it just really wasn't going well. So I went to Steve and I said, I'd just really like to have another project. And he said, well, what do you want to work on? And I said, on viral entry. And so he said, well, I have this project on herpes viruses. And it wasn't anything that he was working on. It was actually a project that was pursued by the late Don Wiley. And so Steve was, after Don's demise, Steve was persuaded to take over this NIHR1. And so he wanted someone to work on it. And so he offered it to me, and I didn't really know anything about herpes viruses, but I started reading about them, and I just thought that they were really cool, partly because so little was known about them. And at the time when he offered it to me, he basically said, if you're welcome to take it with you when you leave. But at that time, I didn't really think about it. I just wanted something to work. But once a herpes virologist, I find always a herpes virologist. Yeah, you can just never get away from them. So,
and of course, that's
probably never will.
For the rest of your career, then you've continued to work on herpes viruses. So it's interesting how decisions are made about what we work on. Mm-hmm. Oh, yeah, I have this project. You should try it, right?
Yeah,
that's the way it works. So then after that, you came to Tufts, is that right, as a faculty?
Yeah.
And you've been here ever since.
That's right.
Early in your career, when you got interested in science, what was the catalyst for that? Do you remember? Was there something that a teacher, or was it in your family?
No, I wouldn't say that it was in my family, but I think I was sort of interested in science. I wouldn't say that I was interested in science. I didn't have really a lot of science exposure until I went to the university. But when I was in high school, during that time, we sort of started learning a lot about genetics, about this great strides in genetics that were done in Russia in 1930s, and then how that was just 20s and 30s, I should say, and then how that was brutally squashed. And so it was something that I just read about, and I thought, oh, that was just so exciting. And also I read some articles about biotechnology. And so I just thought that like all of these things, just everything biology related sounded very exciting. But I don't think that I really knew what science was about, anything about the scientific method. But when I went to the university, so there are lots of opportunities. So sort of as even as a first-year student, you were highly encouraged to basically start doing research in some lab. And so I started doing that. It was actually my first research project that I did kind of on the side, between classes, was on high-temperature superconductors. Yeah.
Which you never went back to.
No.
Right.
No.
Sean, where are you from?
I'm from a town called Leeds in England.
Leeds. Now, was there a concert recorded there live at Leeds?
There was, by The Who.
The Who, right?
Yes, yes. Many, many years ago.
Before you were born?
No, actually, I was alive. But,
Where is Leeds? Is it south of London, west, east?
It's 200 miles north of London.
North. It's not Yorkshire yet, though, right?
It is Yorkshire.
No, because you have the Jeff Almond accent.
Well, Jeff was my mentor as a graduate student.
So you grew up in Leeds, you went to high school in Leeds?
Yes.
And you went to college?
So I did my undergraduate degree at the University of Birmingham, which is about 100 miles south of Leeds, southwest actually, a little bit closer to London. And my undergraduate degree was in microbiology and biochemistry. It was the first year was a pretty course-intensive year, and then after the first year, it was as much focus on research time in the lab as it was didactic classes. And by the third year, it was virtually all lab time. We had, I think, 4 one-hour classes a week, and then the rest of the time you were ensconced in a lab.
So you were a science major?
Yes. Yeah.
Did you enter being a science major?
Yes.
Did you yeah. And where did that come from for you?
So actually, as a kid, I was sort of interested in microbiology. It's sort of weird. Why? There were 2 overriding reasons. One is I discovered yeast could make beer and wine. And the other is that I realized microbes could kill things. And so my first introduction to that was actually realizing that a fungus killed all the potatoes in Ireland and caused the potato famine. And so it fascinated me that these small things could have such a huge impact on humans without even at the time realizing about necessarily human disease caused by some of these microbes. So that was sort of my earliest recollection of microbiology.
So when you went to college, did you have going for a PhD already in mind, or did that come up?
No. In fact, I was convinced that this really is my alternative career. I was convinced that what I really wanted to do was be a physician. So I had applied to medical school, but wasn't accepted in a medical school, but I was accepted into biology programs. And so I started in this microbiology and biochemistry program in Birmingham that I loved and never looked back from.
And that was not with Jeff Almond, right?
No.
Jeff Almond was a postdoc.
No. So
I should let you answer the question.
Sure. So how do I get from the University of Birmingham to Jeff Almond? Well, I realized in the final year of my undergraduate degree that really the practice of science was something that I loved and enjoyed. And so I wanted to do a PhD. I didn't know what my future career would be after that stage, but I knew that I needed a PhD, whether it was going to be to work in industry or academia or government. And so I was fascinated by virology. And I actually applied to work with a guy called Tony Minson at the University of Cambridge on herpes viruses. And I remember actually interviewing with Tony, and at the time it was still legal to smoke in buildings in England. And Tony's office was a glass cubicle in his lab, and Tony was a smoker. and the glass cubicle was absolutely full of smoke. And there was no way I could survive a PhD under such circumstances. I subsequently took up smoking. No, I'm joking. But shortly thereafter, I interviewed with Jeff Armand, and in contrast, Jeff was the youngest professor in the UK and worked on poliovirus and was very interested in trying to use the poliovirus vaccine strains, which had been very successfully used to control poliovirus, but to see if there were other possible uses of those vaccine strains. And his lab had an ongoing project where they'd been expressing antigenic determinants of HIV on the surface of polio and had been able to produce some neutralizing antibodies, clearly not sufficient to lead to vaccine purposes. But my project as a graduate student was to try and see what other functional determinants we could put onto the surface of polio.
And so at the time he was at Reading or Leicester?
Yeah, the University of Reading,
right?
yeah. So my goal was really to see if I could get poliovirus to infect cells via using a different receptor. And so I was replacing the BC loop of VP1, which I'm sure you know and love very well, but with initially peptide hormones to see if we could get them to bind to the relevant peptide hormone receptor. And none of this worked. And I was pulling my hair out, and I turned to herpes viruses again, actually, and to Epstein-Barr virus, because the work of Glenn Namoro had defined a 9 amino acid peptide in GP350, which was sufficient to bind to the EBV receptor and to allow molecules to get taken up into cells following engagement at the EBV receptor. In fact, I still remember the sequence of that peptide. But anyway, I put this onto the surface of polio, and I could, I was so excited I could get the virus to stick to BHK cells that we'd engineered to express the EBV receptor, CR2. And they didn't infect. And I could take the RNA from the virus, put it into the BHK cells, out would come infectious poliovirus. So something was missing. And actually about that time, you published a paper where you'd taken a soluble poliovirus receptor and selected soluble poliovirus receptor mutants. And really sort of the emerging picture of entry for poliovirus was it bound to the PVR, but this was a destabilizing interaction that allowed conformational rearrangements in the capsid and then the release of the RNA into the cytoplasm. So I wrote to you and asked, in fact, for some soluble poliovirus receptor, and graciously you sent some to us.
Glad I sent it. I thought you were going to say I never answered.
No, you indeed sent us some soluble poliovirus receptor, or what I thought was soluble poliovirus receptor. But it turns out what you sent us was solubilized poliovirus receptor from the insect cells. And so I got this material and I got my experiment all set up, and I added the solubilized poliovirus receptor to my cells, and the cells all solubilized.
Yeah, because there was detergent in it, right?
Of course, yes. But in the end, I was not successful in getting the virus to infect, even though it was stuck and anchored to the surface of cells with this EBV peptide. And similarly, with efforts in trying to use bits of GP160 and CD4, we were not able to move beyond the attachment phase into the
Well, as you said, you need a catalytic conversion by the receptor, and that was...
We didn't know that at the time, in my defense.
Yeah, it's fine, it's fine.
So how were you able to graduate if nothing was working?
It's a very good question, Katya.
Did you publish any of this? You must have. Right?
Yeah, there is actually a paper somewhere in...
I found one from Jeff Almond.
Yes, yeah. Somewhere in the archives. Actually, my first manuscript that I ever published was published on VSV before it was on polio.
Really?
Yeah. Yes.
But you finished in Jeff's lab and then you did a postdoc, right?
Yes.
Came to the U.S.
I did, and again, it was a case of, I was actually supposed to go do a postdoc in London on HIV with Robin Weiss, again related to receptor biology, but it was my turn in the lab to do journal club, and I was sort of reluctant to do journal club because I was working on writing up my thesis and trying to finish up and move on. And so I took the path of least resistance, and I went to the library and picked up Cell Science and Nature, and I opened up Cell. And one of the first papers I saw in that was a issue of Cell was a paper from Gail Wirtz's lab at the University of Alabama in Birmingham. And the paper was entitled Infectious Defective Interfering Particles of VSV from Transcripts of a cDNA Clone. And this was 1992, and it was important because it had been really difficult to manipulate the genomes of negative-strand RNA viruses. There had been some advances, the most notable being on flu from the lab of Peter Palese in 1989, but even that was limited to a single segment of flu. And so really the entire field had sort of struggled to get to a point that you'd achieved for poliovirus in 1981, and that before that.
Right here in Boston.
Yeah, just across the river in Cambridge, actually.
That's right, Cambridge. Yeah.
But I mean, that ability to manipulate RNA genomes had been developed for phage a couple of years before that. But the negative-strand viruses still, a decade later, were really still behind. And the opportunities to, if what I realized when I read the paper was, finally, the opportunities are now available, and why on earth was there not infectious virus? These were defective interfering particles, so why don't we have virus? And so I wrote to Gail Wirtz at the suggestion of Jeff, actually, and said, you know, this is exciting, you can do all these different experiments, and Gail offered me a job, and I thought, Birmingham, Alabama, London, England, yeah, why not? And that's how I started working on VSV. Clearly
had never been to Birmingham, Alabama. Is anyone from Birmingham? I don't want to insult you.
I enjoyed it.
I'm sure it's a good place to be for a few years.
Yeah.
And then I want to talk about some of that work in a bit. And then after that, you took your job here, and you've been here ever since, right?
Yes.
Continuing to work on VSV.
Yes, and still, in fact, some of the very questions that I wrote in that letter to Gail.
All right, we'll come back to that. Let's go back to Katya. I want to talk a little bit about your work on herpes viruses, which here's what you write on your web.
Not high-temperature superconductors.
Sure, we could wing it. Why not? I just think the audience doesn't want to hear about that.
All right, fair enough.
We on your website, it's written, our favorite viruses are herpes viruses because they are ancient, marvelously complex viruses that are also important human pathogens. So I had written a question here. How did you get turned on to herpes viruses? Which you already answered. That's
by accident.
Steve Harrison said you should work on these, but in retrospect.
No, he asked me what I wanted. He said, here's a project that you could work on. I said, why not?
Yeah. All right, now I've picked a couple of your papers, and let's go through them figure by figure. No, just kidding. I just talk about the big picture. First one I picked, you're a first author in Steve's lab, 2006, the crystal structure of glycoprotein B. Was this something that he said you should work on, GB?
So that was, so he basically offered me to work on any glycoproteins that were part of that R01. And then I continued Don's collaboration with the virology group at UPenn, so Gary Cohen, whom you know, and Ross Eisenberg. And so I basically, just as a postdoc, it was just really great opportunity because I collaborated with them essentially as an investigator. I mean, I would occasionally update Steve on what was going on, but he gave me a lot of freedom. So that was really wonderful. And we basically, at that time, they thought that GH and GL were more important. And so they sent me some proteins that they purified, and it turned out that GHGL just really wasn't not a good shape, just that complex was aggregated, was falling apart. But they also sent me just a little bit of GB. And it was something happened, like it was coming, like somehow it got stuck at customs. I guess they sent it to someone else, and then it was coming back. And it was thawed there, just all sorts of bad things happened to it. And the amount was very, very small. But I thought, why not? You know, I can just set up a few crystal screens. And I set it up, and and at first I didn't see anything. And then sort of like a month later or 2 months later, I was just sort of going, I think it was like Friday afternoon, and I was going through all these different crystallization plates, and then I saw crystals there. And I thought, wow, this is how cool, but how can I reproduce them? I have no more protein left. But so I contacted them and I said, bad news is that, GHGL that you sent is not good. But good news is that GB, I got crystals of it. And, I hope that that's okay with you. And I said, well, yeah, we think GB is also important. So, that's okay. Yeah, and so then just they sent me more protein, and after a lot of struggle, I was able to determine the crystal structure. So
tell us what, in the herpes particle, I know there are lots of glycoproteins, and what is GB and what does it do?
So what was transformative about that paper that we published was that from the structure, we were able to identify that protein as the viral fusogen. And so all enveloped viruses enter cells by fusing their membrane with the membrane of the cell. And membrane fusion, as you probably know, is a thermodynamically favorable process, but it has a very large kinetic barrier. And so it basically needs a catalyst to overcome that kinetic barrier. So think to your PChem. So hopefully that will make sense. And so, so each enveloped virus encodes several copies of a fusogen. So that's the catalyst that basically fuses the membranes. And so, and in the vast majority of enveloped viruses, there's typically just one glycoprotein, and that performs the fusogenic function, typically in addition to other functions such as receptor binding. But what was confusing about herpes virus is that they have so many different types of glycoproteins on their surface. And by the time that I started working on them, we already knew that in herpes simplex virus, which is what I worked on, there are 4 glycoproteins that were essential for entry. But we knew that one of them, GD, was the receptor binding protein. But we didn't know what was going on with regard to fusion. And there are three other proteins, and any one of them could, in principle, mediate fusion. And in fact, at that time in the field, there's this idea that all 3 proteins were somehow involved in fusion. But we didn't know what was the actual catalyst. And so when I determined the structure, and it was also sort of, from the structure, you couldn't quite obviously tell that it was a fusogen. But around the same time, another group, the group of Felix Ray at Pasteur, determined the structure of glycoprotein G from VSV. And so in that case, they knew that that was a fusogen. And so basically, it just so happened that Felix contacted Steve and just sent him some preliminary figures of the new structure. And Steve and I were actually meeting to look at the GB structure. And then all of a sudden it just struck us that they looked so similar. So then we thought, oh, gee, well, does this mean that GB is a fusogen? So that was really exciting.
So the fusion of herpes occurs at the cell surface, is that correct?
Yes and no. In some cells it does, but actually most cells are entered by endocytosis, and it's quite a messy story, because it appears that different cells are entered by different routes, which we're just only beginning to.
Only a herpes virologist could love this,
Yeah, and so it's just the entry is just it's complex. There's just lots of mysteries.
So when it enters by endocytosis, is there a low pH trigger for fusion, or is it not?
So it's also a contentious issue. It doesn't appear to, so low pH is not required to trigger fusion, because fusion can occur at the plasma membrane at neutral pH, and it can also occur if you just express these 4 glycoproteins along with the receptor in cells. So low pH is not required. So at the same time, the virus can enter by endocytic pathway that is pH dependent. So we still don't know. We still don't know what role pH plays, if any. I mean, I personally don't think that it's important for triggering, but we don't really know.
But GB has a fusion peptide or a fusion?
Yeah, it has 2.
Yeah. Is it in the structure that you solved? Is it hidden or is it exposed?
No. So the structure that I determined represents the post-fusion form. So the inactive sort of final form. Do
you know what triggers the exposure of the fusion peptide of GB?
No, so we also don't know that. So we know that these other glycoproteins, so they play an important role in that, but we don't know exactly what they do. So what we currently think is that, and that comes from the work of many investigators, is that the first event that occurs is that GD, the receptor binding protein, binds the receptor. And then that interaction somehow signals to this GHGL complex, which we have no idea what it's doing, except that it's very important for fusion. And then we think that GHGL signals to GB and activates it somehow. But again, we don't know how. So it's still a rather murky area, but we're sort of trying to
so this GH, you also solved the crystal structure of that.
Yeah,
right?
so that was here at Tufts, actually.
And does that provide any insight to what it's doing in the entry?
Well, it told us that it wasn't a fusogen.
Okay.
No, but actually that was important because for the longest time, people were thinking that perhaps that since herpes viruses are so complex, sort of nothing is, what's a good way to put it? You can't put anything past them. So they could have two fusogens. And we just thought that didn't quite make sense. Why would there be two fusogens? Because they don't have redundant functions. They clearly, both GHGL and GB are required. And so when we determined the structure of GHGL, we could say with confidence that it wasn't a fusogen. And so we proposed that instead it was a regulator.
I have to interrupt with a short story. I was on study section for many years, and I was always surrounded by herpes virologists at the table. There was me, 1 or 2 RNA virologists, and then 5 or 6. And they would say, well, herpes is so complicated. We need a lot of money to figure out. And they often prevailed, in fact. As you say, it's very complicated. Now, in the crystal structure of this GHGL, you say the crystal structure of the fusion regulator complex. So it somehow is regulating fusion mediated by GB, right?
Correct. But so the operational word is somehow.
Somehow. You don't know how exactly.
We don't know how it does it.
Are you interested in figuring that out at some point?
Oh, yeah. We're pursuing it. I mean, it's just really, it's difficult. Well, I mean, I know, I sound just like all of these other herpes virologists.
Need a lot of money, right?
That's right, yeah. I mean, one of the problems is that no one has really been able to observe the interaction between GHGL and GB. And so the interaction has been inferred from some antibody inhibition studies, but it hasn't ever been observed. And so presumably, I mean, you would assume that they have to interact to be able to, for GHGL to be able to trigger GB. Yeah.
Aren't you glad you didn't work with Tony Minton?
Minson. Yeah. It's the code of the herpes virologists.
There is a recent paper you published, which you might be interested in because it uses VSV. You actually pseudotyped VSV with all these 4 herpes? With
this 4, right.
All at the same time, right? Or just separately?
Right. So we actually, so I initiated a collaboration with Sean because of course he did so much with VSV.
Okay. I didn't know that.
And so what he proposed is that instead of just pseudotyping this VSV in trans, so he said, why don't we just why don't we help you basically put this herpes virus glycoprotein genes into the genome? And so that worked with GB. But, and we actually wanted to use this HAP1 cells to basically map out the routes. And so we put in GB, and that seemed to be fine, but of course we need the other ones. And when we put in GH and GL, my impression is that the RNA polymerase from VSV just said, well, I don't know what it said, but basically it was not happy. It was not, yeah.
This is a family show, you can't repeat it.
Yes, yeah. I mean, it said no, thank you, and that's kind of where our collaboration sort of stalled. No, we're still trying, but I think every time we try something new,
but this paper, which was a recent JV paper, the title sounds like you got all 4 of them in the pseudo, and that's correct, right?
But that was pseudotyped in trans.
Okay.
So it's not done.
And so, what does this pseudotyped VSV have? Properties that you would expect from having all these 4 herpes glycoproteins in it?
Yeah, so it basically, we're able to show that it can enter cells in the absence of all these other glycoproteins because HSV has 12 glycoproteins. And so without the 8, so these 4 are sufficient. So that was the first time we were able to show that these 4 glycoproteins are sufficient for entry. Prior to that, people showed that they were sufficient for cell-cell fusion. But as many virologists mentioned, cell-cell fusion and viral entry are not the same events.
So I also noticed that you're interested in tegument proteins.
Oh boy. Am I allowed to use the word complex and difficult or no?
You can say it. Yeah, it's fine. There's no problem. Maybe, you know, that could help get more money if you say things are complex. But tegument, yeah. And you have a 2017 paper, Crystal Structure of UL37, which is one of these. Tegument is the stuff inside the virus particle, right?
Correct. So it's on the outside of the capsid and underneath the envelope.
Right. And, you know, I've been in virology since the 70s, and in the 70s they just said there's something here, we don't know what it is. And I noticed crystal structure. So do you continue to use X-ray crystallography? Have you done any cryo-EM in your laboratory?
Yes, we do. We're still using a lot of crystallography because that's something that we're very familiar with. Cryo-EM is something that we're learning, so we really want to use it, but it's a steep learning curve, and it has been difficult. But that's something that we're pursuing.
Need expensive cryo-EM machines.
Yeah, the tegument proteins, most of them are just. I don't think they're large enough to be amenable, and also presumably asymmetric. So they might not be amenable to cryo-EM, but that's definitely something we want to have as a tool that we can just go to whenever we need to.
Are you excited that the Nobel Prizes in chemistry were for cryo-EM this year?
Yeah.
It's pretty cool, right? Developing of the methods. There's a really nice summary at the Nobel website about that. You know, you could get advanced and beginner summaries of the Nobel Prize. And the advanced one is really good. It goes through the whole history of EM, crystallography, and development of cryo-EM. It's very good. And they talk about the range of, I think 30 or 40 kilodaltons is the lower limit for, they say, for cryo-EM, for getting good structures. The other thing you do, which I think is really cool, is how herpes gets out of the cell. It replicates and assembles the capsid in the nucleus, right? So you're interested in how it gets out. Tell us a little about that.
Yes, it's kind of interesting because we really focus on how herpes viruses get in and how they get out, but we sort of ignore what happens in between those events. Maybe at some point we'll get to that once we figure out the tegument. Yeah, so herpes viruses are very weird when it comes to their egress because they actually bud twice, but they acquire only one envelope. And that's because, so it's like if you look at we're very interested in how herpes viruses manipulate membranes. And so if you look at the whole life cycle of the herpes virus, it's basically, it's fuse, bud, and then repeat, fuse and bud. So you do this. So basically capsids are assembled in the nucleus, as you said, but they're too large to get through the pores. So they have to get out by some other mechanism. And so what the virus has come upon, discovered, evolved somehow, and actually somehow is also a very interesting part that maybe we can talk about. So basically what it does, so these capsids bud into the inner nuclear membrane, form these perinuclear viral particles that really don't look anything like mature virions. So we know that they're sort of these intermediates.
There's no glycoproteins in those, right?
Presumably they have glycoproteins, but it's not clear whether those are important for fusion. So that is something that we really don't know. And then they fuse with the outer nuclear membrane.
So they would need glycoproteins to do that, right?
They need a fusogen. Yeah. But what they use as a fusogen, we don't know, whether it's glycoproteins. And then they basically end up, so in terms of morphogenesis, it's sort of like it's a net zero event. So they bud and then they fuse, but they do accomplish getting out of the nucleus.
Now you have a naked capsid in the cytosol, right?
Naked capsids, they may be already coated with some tegument proteins, and then they travel to these vesicles derived from trans-Golgi network or endosomes where they undergo envelopment.
So they bud into the
So they bud into these vesicles.
And then they exocytose out of the Golgi,
correct.
Right? And they get a second envelope.
No, it's just one. So they basically bud into this, into what will become exocytic vesicles, and then these vesicles fuse with the plasma membrane.
By exocytosis. Yeah. So I love to give this question on my virology course exam. What's the origin of the final herpes membrane? Where does it come from in the cell, the one that's on virus particles?
So that depends on whom you ask, but it's trans-Golgi network or endosomes, or both, because the vesicles have...
I'm not going to give the answer because they'll listen and get it for next year.
Yeah.
But it's a good thought question, and they have to draw it out and think about it.
Well, I mean, it's if you want to know, so during herpes virus infection, the Golgi, trans-Golgi becomes dispersed, so you really don't have these organelles, but what you have are these vesicles that bear markers of endosomes and TGN, so that's where they butt.
Do all the herpes viruses do this nuclear egress?
Yes, it's a conserved mechanism because they all have large capsids. Right. And so I used to say that, I think I wrote some review, and I said that herpes viruses are the only viruses that do this. And then actually someone emailed me, I don't remember the name of this researcher, but he said, well, herpes viruses are the only animal viruses known to do this. But they're actually insect viruses that mediate this process. And so when I wrote the next review, I made sure to reference.
Yeah. I was going to ask you, there must be other large nuclear viruses that have to do the same thing.
Viruses that infect insects.
Including ones we don't know about.
Right.
Really big ones. So are there specific questions you're trying to sort out about this egress?
Yeah, so we're trying to figure out what molecules are involved in this process, and as you know, so kind of our major contribution to this area was determining that these two viral proteins that form a complex that's important for nuclear egress, that they in fact represent the budding machinery of the virus. So basically what we showed was that when we expressed these two proteins in E. coli, we purified the complex and added it to synthetic membranes, we saw just this crazy amount of fusion that occurred very rapidly. So clearly showing that this is what these proteins do in the virus. But now the challenge is because normally during viral infection, you don't see this crazy budding. The virus is clearly controlling this. So now what we're trying to figure out is how it is controlling this, how it's keeping it repressed until the capsid comes along, and how the capsid activates it. Yeah, so that's something we know.
Whenever I hear egress, I always think of the P.T. Barnum story. Do you know that? No. Do you know that one, Sean? So P.T. Barnum, of course, ran a circus, right? Barnum and Bailey circus. And he was a clever fellow, and once he put above the door in the circus, egress this way, and everyone thought it was an animal, so they went. And of course they left. It was his way of getting people out of the circus. And I think the same way with herpes virus. Here's the egress, here's the way to get out.
I actually don't know why it's called egress, but it's a term that I mostly encountered in herpes virology. I think that herpes virologists just really want to be different.
I think every virus field, they have their names and they stick. The tegument is a weird name, right?
I know. Yeah. I actually don't know the origin of it.
And it's for someone saw it on an EM one day, which reminds me, this is a herpes virus, a giant microbe that you gave me. What is this red thing around it? It looks like a halo of...
So this is a negative staining artifact.
Artifact, right?
Yeah. Yeah. So it's so it comes. So the giant microbe toy is basically inspired by this very popular images from the 1980s, I think, that were taken by Linda Stannard. So she's a very talented, probably still is a very talented electron microscopist. And what they show are this, so it's the image of the, I think it's HSV virion. And so it has its capsid. You can see the capsid very clearly. And then outside of the capsid, you kind of see, like, it really looks like, the capsid looks like a yolk. So like imagine like you break the egg into a pan, right? So the yolk is the capsid and the white is basically, the outline. And so if you really think about it, what this is that the viral envelope has gotten disrupted by the negative stain, and that's actually the reason why you see the capsid, because the negative stain got to the capsid, and now you can see it. But I think it was just a very photogenic. I mean, the image was really really nice.
Like you said, they took the EM, which is an artifact, and they thought this is what it looks like. But we need more science education out there, because this. It's not herpes, obviously. But someone thinks it is a giant microbes.
Well, it is, it's just not intact.
It's not intact, right? I mean, I think the capsid would be cool to have a giant microbe, right? Yeah. Here's what they say here. This echoes what you have told us.
Okay, thank goodness.
One of the main types, one of the main types causing cold sores is herpes simplex virus type 1, but it's anything but simple.
That's right, and we need a lot of money to figure out how
We need
it
lots of
works.
money. That's it. Lots of money. And you know, the RNA virologists at the study sections would argue, why do you need so much money? To no avail. We'll get back to you. I have some more questions, but I want to talk to Sean a little bit. Now, at the time you went to the Wirtz lab, had there been an infectious clone of a negative strand RNA virus yet?
No.
The rabies came out later.
Yeah.
So were you the first to do it?
So the first lab to rescue a non-segmented negative strand RNA virus from a DNA clone was Klaus Konzelmann with Herpes Virus. In fact, it was the work of Matthias Schnell, who's in Philadelphia now. And then the group of Jack Rose and the group of Gail Wirtz published close to each other, although Jack's paper came out slightly, while ours was actually accepted but not yet published. Both were published in PNAS. For VSV, yes. So Lawson was the first author on Jack's paper, and I was the first author on the one from Gail's lab. So 1995, and then at the end of 95, it was successfully accomplished for RSV and Sendai, I believe. And then shortly thereafter, there were a whole bunch of other reports of rescue of negative strand virus.
So it must have been very exciting for you to do this.
Yeah,
right
it was great.
at the start, right? What was the key to getting it to work? I know people tried for years, right?
Yeah, so
the key is really weird, isn't it?
Yeah, the key is weird. So, I mean, it might seem obvious now, but so the key was to make the antigenome instead of the genome in the cell. So one of the major contributions that Peter Palaise's group made was the notion that you have to reassemble the ribonuclear protein complex, because that's one of the things that's unique about the negative strand RNA viruses. The negative sense RNA, and they have to be completely coded by this nuclear capsid protein and associated with a polymerase in order to initiate infection. And so what you're trying to assemble in the cell is the ribonuclear protein complex. Peter's lab did it first in vitro for a segment of flu and transfected that RNP into cells and superinfected with flu, and were able to get that RNP to be incorporated into flu, but it was rapidly lost during passage. But for the non-segmented guys, efforts had focused on reconstituting that RNP from expressed plasmids, from proteins expressed from plasmids, and the genomic RNA in a cell. And so what you're doing is you're expressing the 1.5 kilobase N message, the 0.8 kilobase P message, and the 6.5 kilobase L message as positive sense transcripts by T7, and the 11.2 kilobase genomic RNA that's negative sense. And
that's what
they
happened.
can hybridize.
Just hybridize.
Yeah. So, of course, this was a significant problem. For VSV, it turns out that there was another problem. T7 RNA polymerase specifically recognizes the 23 nucleotide sequences conserved at every junction in the viral genome and terminates. So you're trying to make this 11.2 kilobase piece of RNA, and the transcriptase that you're using to make it in the cell that's expressed from a recombinant vaccinia virus is just, it's not going to do it. So there were two problems. So Klaus's lab had used the antigenome and expressed the N, P, and L proteins of Herpes virus, and that assembled an RNP, and the polymerase that was expressed from the P and L was sufficient to copy that then into an encapsulated negative sense RNA, and out came Herpes virus. And in fact, we learned of this at the International Negative Strand RNA Virus meeting in 1994. And on discovering this, I think everybody in the field went back to their labs and
got it to work. Yeah.
Spent, well, it took a number of years for some people, but I think it was the the main focus of every group that was trying to do genetic manipulation of negative strand viruses. Let's get an infectious clone.
Yeah. So Andy Ball also contributed something to making the end precise, right?
That's absolutely right. So that was the trick to actually getting the cDNA. It turns out you have to have both ends correct. The 5' end of the transcript is a little bit more forgiving, and that's dictated by the initiation by T7 RNA polymerase. But the 3' end has to absolutely be perfect. It has to be the authentic 3' end. And so the way that the contribution, Andy made many contributions, but one of the important contributions that he made here was to introduce the hepatitis delta virus self-cleaving ribozyme into the plasmid so that the T7 transcript would be cleaved to generate the precise 3' terminus of the genomic RNA. It's not quite the precise 3' terminus because the cleavage by the ribozyme leaves a cyclic phosphate instead of a 3' hydroxyl, but it turns out that doesn't matter.
Good enough.
Yeah.
And is that something he had done first with DI RNAs?
Yes, that was how it was.
And then Konzelman adopted that to his rabies.
So the rabies clone.
Right.
So you needed both the plus sense and the authentic end.
This may sound like inside baseball, but it's really cool history because his whole career has been based on this discovery, pretty much. Would you agree with that? Almost all of it?
Yeah, I would say. Yeah.
I mean, he continues to do VSV, and the pseudotyping that was done, it depends on this system. So it's really cool. And I lived through this. I was watching everyone trying to make infectious negative strand RNA clones, and they couldn't do it. And this turned out to be a key. So it's really interesting. So you published so many papers as a postdoc subsequently using this to look at different aspects of RNA synthesis, right?
Yeah. So I mean, what do you do when you've rescued the virus from DNA? Well, you can do whatever you want, right? So, you know, we made pseudotypes. We shuffled the order of the genes of the virus. We mapped all the requirements in the promoter elements for RNA synthesis. We made viruses that express fluorescent proteins. In fact, there's so many things that we did that still remain in my lab book and were never published. It's embarrassing. But a fair number of them did end up being published. And one of the things that I actually had been fascinated by was regulation of viral gene expression. And there had been this, speaking of Andy Ball, actually, Andy had been one of the people, in fact, was the first person who used the approach of ultraviolet irradiation of the viral genome to determine the order of the genes of VSV. And in fact, the way that works is UV irradiation induces, we presume, covalent dimers between adjacent U residues, and that blocks polymerase progression as it moves down the genome. And the way that these viral mRNAs are made from this single piece of RNA is that they're transcribed sequentially. So the polymerase has to load at the 3' end and then start at the beginning of the first gene, which turns out to be N, and end at the end of the first gene, and then restart at the beginning of the second gene. And so what that means is that the physical target size of any gene in the genome is the sum of its own physical target size plus all the genes upstream, because the polymerase has to walk through it. Now, the one unresolved issue was this little promoter element at the 3' end, which was the 50 nucleotide leader region, and whether or not you had to copy that before you copied the first message. And one of the reasons that was an important thing to resolve was during replication, the polymerase absolutely has to initiate at the 3' end. And so how do you discriminate as a polymerase between the processes of replication and transcription? Well, you could initiate internally and then you would become a transcriptase, or you could initiate at the 3' end, or at some point during transit through the first part of the genome, a commitment step has to be made to undergo replication or transcription. Or there are other hypotheses. You could have two different pools of polymerase. But what I realized was now we could manipulate the genome. We could actually use the same UV mapping experiment and look for things that would tell us whether or not the leader region had to be copied before the first gene. And I did this, and I first started out by trying to put small little genes between leader and N, and this process did not work. Turns out that a message has to be a minimum size before the VS3 polymerase will actually initiate and terminate. And so that was sort of a clue also that what was going on at the 3' end of the genome was different. And in the end, the way that I solved this question was by changing the ability of covalent U dimers to form in the leader region in response to UV light. And so the end result of that was that the polymerase could actually initiate internally, or that the manipulating the that the UV target size of the first transcript that was made from the genome did not depend upon copying the long.
And this was, you did this in Gail Wertz's lab, right? Yeah. This paper, by the way, this UV mapping, I always used to teach it. And it's a classic, 1976, Ball and White. Yes. And so a year ago on TWiV, we decided to do this, do some old classic papers, right? And I wrote to Andy, I said, give us some historical context, just a few sentences. So he sent us a 5-page referenced document telling the story. It's published on our site. It is just fascinating to hear how the whole thing emerged and what led up to it. It's just great. But I said, did you write this for a review article or something? He said, no, I just whipped it off in a few minutes. Because he remembered it all. This is the stuff that is really great that I love to hear.
Yeah.
What would you say, you still work, when you came to Harvard, you still continue to work on aspects of VSV RNA synthesis. Is there something that's unsolved?
Oh, there are lots of questions that are unsolved. I mean, the reason we work on VSV is it's the only member of the entire order of non-segmented negative strand RNA viruses that authentic transcription can be reconstituted in vitro from purified components. So there are a huge number of important pathogens in this order. And we know that polymerases and RNA synthesis in general are terrific targets for the development of antiviral drugs. But there are many questions. One of the questions that was unsolved when I started my lab was, and in fact it was the focus on which I started my lab, was to really push towards getting a better structural and functional understanding of the catalytic component of the replication machinery, the large polymerase protein L. And this is a 250 kilodalton protein. It has an RNA-dependent RNA polymerase activity. And then in addition to that, it has all of the enzymatic activities necessary to add a cap structure, an mRNA cap structure, to each of the viral transcripts. And we've known, in fact, since the discovery of the cap, which was partly because of studies. In fact, the cap was discovered through studies of vaccinia virus and reovirus, and so viruses in general, but it's been known ever since the discovery of the cap that the way that VSV forms its cap is through a different mechanism. But precisely how that, mechanistically how that worked, wasn't appreciated until 2007, 2006, 2007, in a paper that actually came out from the group of Omio Banerjee, and in particular Tomo Ogino, who now has his own group at the Cleveland Clinic, who demonstrated that in contrast to other capping reactions, what happens is, in conventional capping, you form a covalent intermediate between an RNA guanylyl transferase and the guanosine monophosphate, and you transfer that onto the nascent mRNA. But in the case of VSV, what happens is you form a covalent adduct between a monophosphate RNA and transfer that onto an guanylate acceptor. It's a completely different reaction. And we defined in the L protein a motif that's present in this conserved throughout all of the non-segmented negative strand viruses, that when you modified that motif, capping was abolished. And so we've been very interested in both the structure and function, and we've been trying to determine structures of L using crystallography initially. without any success. And over the years, a number of people in my lab tried expression of pieces of L and all sorts of fragments and approaches. And we got very excited, actually, about 2009 when, together with Tom Waltz, we did a negative stain electron microscopy study of VSVL and were able to generate some first images of really what the protein looked like. And what it looked like was a donut with 3 globular domains appended to the donut. And if you think about polymerases, then something has to go through the center of the molecule. And so we made the assumption that the donut was the RDRP and that the globular domains represented the capping enzymes and the cap methylation enzymes. And so we sliced and diced the protein and defined, in fact, that the donut really did contain the part of L that was the polymerase. And we obtained data that the methylase domain formed these 2 globules that were appended to the donut. And then by inference, we said that the other globule was the capping enzyme. That turned out to be wrong, actually. It wasn't until, I mean, the methylase being 2 globules and the polymerase being the donut was virtually right, but the capping enzyme was gone. And it wasn't until we, together with Steve Harrison and Nico Grigoriev, did the cryo-EM structure of the VSV polymerase, which we published now a couple of years ago, where we got a 3.8 angstrom structure of the polymerase of VSV. Wasn't until then that we realized that the capping enzymes actually sat right on the back of the polymerase and is intimately connected with it. And in fact, the 3 globules that we saw by negative stain were what we now call a connector domain, and then the methylase domain, and then a structural C-terminal domain. So it's amazing how structure sort of changes your understanding of how things work.
That was a big deal. It was the first polymerase from a mono-nega virales, right?
Yes.
The flu had been done before, segmented negative.
Flu had
right?
the structure of flu polymerase had just come out, which was solved by crystallography by Steve Kusak's group, yes.
So that was, yours was cryo-EM, 3.8 angstroms. And
3.8.
And this was just the L protein.
It was L with a tiny little piece of a protein called P. And what P does is it engages L, and it also engages the N protein that coats the viral RNA. So if you take the L polymerase alone and you give it the N-coded RNA, right, you can't copy it. You have to have P. And in fact, P induces a little bit of a conformational change in L. We got very excited when we looked at it by negative stain EM because we thought, you see a compaction of the globular domains down onto the donut. And we thought, wow, this is a much more uniform complex and we should be able to crystallize this, but it didn't work. Wasn't until cryo-EM that we really could get a good structure.
I was wondering how you continued to work on VSV. You know, there's a big emphasis on clinically relevant virology research. Been so for a long time now. But I guess you put it in terms of other negative stranded viruses that are human pathogens, right? Yeah,
of course. I mean, at one stage during the studies, you know, I'd always maintained that because VSV was the only virus for which you could do something in vitro, at one stage during our studies of VSV, we developed a method so that we could get the polymerase to actually copy RNA in vitro, where the RNA was just a chemically synthesized piece of RNA. So you could at least do some sort of studies of RNA synthesis in vitro. And I thought, oh no, I've now actually done something that allows anybody who can express a polymerase from any negative strand virus to actually study its properties in vitro. And it turns out that, thankfully, that nuclear capsid protein is really important. And without it, the polymerase is not fully processive. It still is engaging the RDRP domain. We don't know how. We'd like to actually know by getting a structure of the polymerase, copying an encapsulated template. But we know that it influences the polymerase in two important ways. It alters the processivity of the polymerase, and it alters the impact of substituted nucleotides on polymerase behavior. So the best example that we've published is one of the ability of the polymerase to incorporate dNTPs instead of NTPs, and how that changes depending on whether the template is encapsulated or not. So it really does alter the RDRP active site.
So about the time you had solved this structure, I remember we were writing the last version of our virology textbook, and I tried to get some figures from you. I don't remember if I ever did. It's like getting soluble polio receptor from me. I don't know what you sent, but we wanted to put it in the textbook. But now we're working on the next edition, so if we didn't get it, I think we didn't put it in, if I recall, because you didn't send it or something. But, but I don't remember. I have another question for you. So you worked on these wonderful plus-strand RNA viruses with Jeff Allman. How could you go to a negative strand, which is so inelegant and inefficient? I mean, a plus strand goes in the cell and boom. In fact, why are there anything other than plus-strand RNA viruses?
Well,
it's a philosophical question.
Yes. I mean, I like the differences between them. Why they exist, we can debate back and forth. I don't think we'll ever get an answer that's suitable to that question. But I like the differences. I like the fact that here is this encapsulated RNA template that has to be copied by polymerase. I like the fact that the replication of these viruses occurs in the cell in structures that aren't bound by a membrane, in contrast to the positive strand viruses. So I think it tells you something about how viruses have solved the same problems throughout evolution, and that there's more than one way to cook an egg, so to speak.
More than one way to cook an egg. Well, I think that's the answer. It works because it can, right?
Yeah.
The different strategies. All right, Katya, let's do some fun questions. Well, these have all been fun, right? If you hadn't been a scientist, what would you have done?
What would I have done?
Yeah, what kind of career would you have had?
I would have become a diplomat.
A diplomat? Really?
Yeah.
And why?
I don't know. It's just, I think that when I was growing up, it's just the idea of traveling to other countries and speaking different languages was so appealing. And when I was growing up, it was really difficult to travel from Russia. And so I just remember that being fascinated with the career of a diplomat.
I think you've told us what you would rather have been, right? A doctor?
It's not what I would rather have been to a scientist. It was what my initial career aspirations.
So what would you rather be if you weren't a scientist?
I think I would be a chef.
A chef?
Yeah.
What would you cook? Pastry, main course?
Oh, no. Not sweet things.
Not sweet things?
Definitely not sweet things.
A main course chef?
Main courses, yeah.
So working in a high-end restaurant?
Oh, yeah.
Do you like to cook?
I love to cook. A good food and wine are the secret to life, in my opinion.
You know about Blue Apron, by any chance?
I do.
Well, they're not a sponsor of this episode, but check them out. Have you tried them?
No.
Okay. You should check them out. I have some free coupons I can give you.
Oh, wonderful.
Katya, what is the single thing you think you have done that has given the most to science as a field?
Well, judging by the number of, like how many papers reference your paper. Mm-hmm. So I think that must be the structure of GB. That's the most cited of my papers.
That was that first structure you did with Steve Harrison.
Yeah.
Okay.
Yeah.
How about you, Sean?
That's always a tough one for me to answer. I mean, you insist on just one. I mean, I would say that there are three things that I would say
as many as you want.
Equally important, and I don't think that not discriminating between them. One, I think, is the recovery of VSV from a cDNA clone that's been used extensively by many labs around the world, and in fact is the basis of an infectious clone of VSV was the basis for developing what is now a vaccine for Ebola. Yeah. And the strategy can be adapted. This is work of Heinz Feldman, actually, not work of ours, but the strategy has been adapted.
Can I add? Making incredible rabies vectors that target neurons, yes, monosynaptic transmission. It's amazing. Yeah. Self-inactivating vectors. They're going to map our brains with your technology.
Well, I mean, the people have been using the rabies technology for that, but there are certainly neurobiology labs that are working with VSV, and we've helped, in fact, locally with Connie Sepko's group at HMS. But then also, I think a pretty important discovery that we made was a collaborative effort between my group and Tyne Brimmelkamp's group and Kartik Chandran and John Dye's 6 years ago now, where we discovered Niemann-Pick C1 as the receptor for Ebola virus. And personally, I really love the fact that we solved the structure of the VSV polymerase, the first structure. It's one of what I hope will be many structures, but if you want to develop sort of inhibitors of polymerases, having structures, I think, is really...
So the receptor and the approach you use is very interesting, which we don't have time to talk about, but I understand you're visiting Colombia next month, so maybe we can sit down and record another.
Sure.
So talk about that. An arc. We'll make an arc of TWiV. All right, Katya, what do you know today that you wish you knew when you were starting your career?
As a graduate student, as a postdoc, as
anytime, a faculty. Just at the beginning, you know.
I don't know. There are just so many things. It just comes up every day. I think, oh, I wish I had training in this, or, you know, I wish I had a degree in psychology. Sometimes I think I wish I had a degree in accounting.
Okay.
I don't know. I wish that, in a way, I wish that I'd known just how hard and science is and how many times you fail before you succeed. But at the same time, I feel like it's best not to know it, because then you might get intimidated by it. So with every new set of experiments, I'm just always so optimistic. I'm thinking, oh, this one's going to work the first time. So, I don't know.
Okay. Sean, do you need me to repeat the question?
No, I wish I'd known how to gene edit human cells.
You know how to do that now?
Well, yeah. But that would have really changed, I think, a number of things that I would have done, because it's been such a transformative technology. And, you know, I mean, essentially, I'm a viral geneticist. So the application of genetic tools to manipulate the host is really the other part of the equation. And I think is really making huge impacts in how we as virologists who are interested in virus-cell interactions function. So I wish that I'd known how to do that at the outset of my career.
Katya, what do you tell a new student who comes to you and wants to be a scientist?
What do I tell them?
You sound forget it.
No, I mean, I just try to get them excited about herpes viruses. I tell them just how rewarding it is to work on them, how intellectually engaging it is, and how much you get to learn, and how many discoveries you get to make. And I think that, yes, I think I sort of try to do that, to get them excited about herpes viruses and the fact that there are just so many discoveries to be made, that it's just a largely uncharted territory.
Do you tell them they're complicated?
I tell them that that's part of the appeal of working with herpes viruses.
How about you, Sean?
I usually tell people that they should get the best training and the education in the fundamentals that they possibly can, and then pursue something that really excites them, because without being creative in your approach to science, you're not going to distinguish yourself from other people doing science. And I think ultimately the best advances come from creative approaches.
But it's not something that you can teach.
No, I know, but you can teach like the basics of a good grounding. And I think that if people are pursuing something that really excites them, then I think you are more likely to come up with a creative solution to a problem.
Yeah, I agree. And just sort of thinking, well, I think it's a cliche, thinking outside the box, but I think it's exciting to get people in the lab who really don't know anything about herpes viruses, because then they're more likely to come up with interesting questions, and even more importantly, to question things that we take for granted. Yeah, I think that's incredibly exciting. I think with every person who joins my lab, we just collectively benefit so much. Definitely, I benefit a lot from that.
Okay, Katya, have you read any good books lately? Anything you want to share with us?
Well, for the last 2 weeks, I've been reading grants because I'm going to a study section next week. But yes, I started reading a very interesting book. Actually, it's written by a colleague of yours at Columbia. It's called Failure by Stuart Firestein. It's a really exciting story about how failure is not just a part of science, but it actually moves science. And he makes an argument in one of the chapters that we should really not just teach students the facts, but teach them how we have arrived at these facts and how long it took, and what were all of the different ideas that sort of didn't turn out to be true. So I've just started reading this book, but I'm enjoying it immensely.
Step 2.
He also has Ignorance.
Ignorance.
Right? Yeah.
I think Ignorance was the first. Yeah, we've had him on TWiV to talk about these 2 books. They're great. They're really good. How about you?
So I've actually been, other than reading scientific articles, I've been reading some potential screenplays that my significant other has been writing. So I haven't really read any books. But they're sort of interesting. One is about the aerobics fad of the 80s.
The aerobics, I remember that. I'm not a big fan of the 80s. I lived through it. In fact, my wife was taking me to the airport today, and she had on some satellite station playing all 80s music, and it just didn't bring back good memories.
Yes, I think the script is not intended to bring back good memories.
Well, hopefully in December you will have read something and you can tell us something else. Katya, what do you do for fun?
What do I do for fun? I like to cook.
You also like to cook.
Yeah. I like to cook. I'm really trying very hard to teach myself new languages, which is it's difficult to find time for. And I really love to travel. So when I have the opportunities, I travel.
What's the last place you went to? Abroad
or domestically? So the last place I went to abroad was Belgium. Yes, I went to a meeting. But the more exotic places that I've been to so far are Japan, Australia, and India. And actually, India is probably the only exotic place that I've been to that wasn't connected with going to a meeting. So actually, so that's something you ask. What I tell new students in the lab, I tell them that science takes you places.
It does,
you get to travel, internationally.
How about you, Sean? What do you do for fun?
So I like to be pretty active. I like to bike, and I love the outdoors. I like hiking. I like skiing, and I was just up in British Columbia actually about a week or so ago. Enjoying the outdoors there with some hiking, and I also like cooking and drinking wine. But
all right, I have one last one. Unfortunately, this is not as much fun as the others, but I think we need to address it. We are in a society now that is increasingly skeptical of science, and in fact we have an administration that seems to be anti-science and not believe science facts. So I think we all love science because it's about facts, and that's it. There's just facts. They're not alternate facts. They're the facts of science. And that's what we like to find. So what do we do about this? You know, our administration is spreading this idea that they don't believe certain aspects of science, whether it be climate change or what else, vaccine effectiveness, so forth. Katya, what can we do to counter this as scientists?
Well, I'm personally very passionate about communicating science. I think it's very important for everyone who is a scientist to be able to explain what they do, not only to their close colleagues, but also to other scientists, and even more importantly, to people who are not scientists, to their parents, to their grandparents, to their significant others, to pretty much everyone. And I think that I really don't buy the argument that the people work on things that are so complicated that it's impossible to explain it to a lay person. I think that if you understand what you work on, you should be able to communicate it at any level. You should be able to communicate it to a 6-year-old or to, my in-laws who are very smart, but they're not in science. And so I routinely have to explain to them what I do using various analogies, sometimes props. And I'm happy to say that they, for the most part, they understand what I do and in turn try to communicate it to their friends. So I think that that's very important.
Yeah, if we all did that, all the scientists, that would really help, I think. How about you, Sean? What can we do?
Yeah, I agree. I think it's public awareness of science, and I think it's public awareness of science at multiple levels. And one is awareness of actually what seemingly science that's funded by NIH that may not seem apparently useful at first glance to somebody who's a member of the public, what the impact of that is. I mean, I think if you think about mapping the promoters of baculovirus, right? Well, how would a grant on mapping the promoters of baculovirus fare at a study section these days even? I mean, it wouldn't, right? But in fact, that's been fundamentally important for a huge amount of what we do for protein expression and also in developing vaccines. And I think communicating to people how the collective advances of science really lead to huge things that impact our lives and impact the bottom line of the country, I think is something that we should focus on too.
Well, of course, that's why we do TWiV. We try and get science out. We have conversations that we want people to listen to, but I also think all of us, and all of you, if you're going to be scientists, I assume you are, or do science in some way, you have to communicate to the general non-science public what it is you're doing. Because if all of us do that, I think it would make an effective voice. There's a good book that we mentioned last week on TWiV by John Udell. He's written a book about, it's called Truth Wins, the great things about biomedical science research. And it's kind of a summation of his career and then what he thinks is going on with science. He has a wonderful summary of how we got to where we are now. So after World War II, people realized that we were victorious because of science and technology. Antimicrobials, all sorts of airplane technology, and many other things were developed by scientific research. And Vannevar Bush went to the president and said, we have to support science. And he's really responsible for making science research the way it is today. And then we lost that. We seemed to get into a point where it wasn't appreciated anymore. And John's book does a good job of recounting this. So go to last week's TWiV show notes, and it's a free download. I highly recommend it because he's very good about getting you excited about science, what's great about it, what doesn't work, and so forth. And for example, he says, cutting the NIH budget is just downright stupid because it does so many great things for society. So check that out. All right, that's TWiV 464. You can find it at microbe.tv/TWiV. You can find it at Apple Podcasts. In fact, if you have a smartphone or a tablet, you can listen to TWiV easily. There are many apps that you can use to listen to the podcast. And if any of you do, please subscribe. That way you get episodes automatically. If you don't subscribe, please subscribe to the shows. Even if you don't listen, just subscribe. Okay? And that really will help us to fund what we do. We love getting questions and comments. TWiV@microbe.tv. And if you like what we do, you could support us financially in very simple ways. You can go to microbe.tv/contribute for ways that you can help us. I want to thank my two guests today for spending an evening here at Tufts University. Katya, Heldwein, thank you so much for joining us.
Thank you. It was a pleasure.
And Sean Whelan from Harvard Medical School. Thank you, Sean.
Thanks. It's a pleasure.
I'm Vincent Racaniello, and you can find me at virology.ws. I'm also on all the other social media, Twitter, PROFVRR, Instagram, PROFVRR. We have a This Week in Virology page on Facebook. I'm on Google+. Follow me somewhere, see what I do, and emulate it. I think it's important that we all do it. I want to thank the Boston Student Postdoc ASM chapter for helping to organize this. In particular, Allison Matthews, thank you for doing all the behind the scenes. And I gave Allison a TWiV mug, and thanks. So enjoy that. Fill it with your favorite beverage. I also want to thank Islam Hussein and his son Adam, who are sitting here. They picked me up from the airport. They helped me set up. Islam and his son do a video virology blog in Arabic called Viral Vlog. Okay, you can find it on YouTube. Unless you speak Arabic, you won't understand it, but you can see you can see the pictures, and they're very nice. So check that out. I want to thank the American Society for Microbiology for their support of TWiV. I also want to thank the sponsor of this show, the Defense Threat Reduction Agency. They help to pay the bills. And the introductory music, which is by Ronald Jenkins. You can find his work at ronaldjenkins.com. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. Thank you.